Welcome, everyone, to the sixth webinar of Obesity 2020, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. Today's webinar is titled Mitochondrial Membrane Lipids and Respiratory Efficiency, and will feature Dr. Katsu Funai, Associate Professor at the Diabetes and Metabolism Research Center at the University of Utah. In his presentation, Katsu will talk about how mitochondrial membrane lipids can affect metabolic efficiency and propensity for obesity-related diseases. This webinar has been sponsored by Aurora Scientific, so a big thanks to them for helping to make this event possible. And with that, I'm very pleased to welcome the speaker, Katsu Funai. Katsu, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Thank you, Liam. I'd like to take the time to thank also the APS leadership, Inside Scientific, and the guys at Aurora Scientific. I also would like to thank everyone for joining in for this webinar. I've listened in to many of these online webinars, including the ones offered through Obesity Webinar Series. And one of the things that really inspired me during this pandemic time is the willingness for the investigators to share some of their unpublished work. And so in that spirit, I wanted to focus my talk today mostly on data that have not been published yet. The central premise of my laboratory is to understand lipid-mediated mechanism that induce obesity-associated metabolic diseases. And today, I wanted to talk to you about the large bulk of the focus in our studies in mitochondria. So one of the questions that we try to think when we try to understand obesity-associated metabolic diseases is what is it about the obesity that is causing these metabolic diseases? And I think one of the previous speakers for this webinar series, Kevin Hall, have illustrated this point really well. But there are many co-manifestations that occur with obesity, such as increase in adiposity, changes in circulating metabolites and cytokines, changes in energy expenditure, changes in energy intake, combining those two, also the concept of energy balance and what happens when there's an energy mismatch between the supply and expenditure. And then it is this last point that I want to focus today, and that's something we think about a lot. What happens to our cells and our systems when the energy that we are taking in and when energy that we are utilizing are not matched? So of many different cellular consequences that may occur when there is an energy mismatch in our body, the focus that we have is the effect that the mismatch have on mitochondria. And as many of you know, mitochondria is an organelle that have an ability to harness energy from substrates that come from food, in a form of high electron and high energy electrons. And these electrons, or the energy from these electrons, undergo several processes that then culminates in the resynthesis of ATP, which is the common currency of the cell. And so when there is an energy mismatch in the cell or two mitochondria, as in the pressure or the flux of electrons that is coming into mitochondria and the ATP demand of the cell, so the pool of the energy burden to mitochondria, when this is mismatched, such that occur with overeating or perhaps a sedentary lifestyle, then there is, again, the mismatch of energy that's coming into energy that's going out. And as you might imagine, in these kind of circumstances, there's a pressure buildup in the mitochondria resulting from this mismatch. And we believe that when there is such mismatch, the mitochondria adapt. And the way that mitochondria adapt, we think, is by turning its machinery into a more inefficient, energy inefficient machinery, such that the pressure that is built from this influx is relieved by a secondary measure, such that the energy is released and there are consequences from such release of energy that it's not going out to produce ATP. So what are we talking about when we say inefficient or energy inefficient mitochondria? We're really talking about mitochondrial oxidative phosphorylation here, and by that it's a series of process that takes place. It's not a process that's done by a single enzyme, but rather by multiple enzymes that spans across the inner mitochondrial membrane. And there are different ways to think about this. The way we think about this is by breaking these processes down into 4 distinct steps, step 1 through 4. So in step 1, we have these high energy electron donors such as NAD, H, and succinate that donate these electrons into complex 1 or 2. And in step 2 is a process whereby these electrons are transferred within these complexes as well as among the complexes to complex 3 and 4. And simultaneously, these complexes utilize this energy from these high energy electrons to pump protons from matrix into inner membrane space. And we call that step 2. Now, the number of protons that's being pumped into the inner membrane space by step 2 does not equal membrane potential. And that is because, one, as shown in this figure, some of the protons can leak back into matrix. And also, the membrane potential is not simply a mathematical sum of the protons pumped in and protons leaking out, but it's also divided by the volume of intermembrane space, because the membrane potential is not near the number of protons, but also how much of that protons is in any particular space. And so that could also be modified by the shape and the size of intermembrane space, and specifically crystals. And finally, in the step 4, the membrane potential, proton membrane potential generated through the step 2 and step 3 is now utilized as a protomotive force to resynthesize ATP. So this is a schematic, but I think we can also think of it this way. And this will help us understand and also quantify the efficiency of oxidative phosphorylation defined by these 4 steps. And so in this schematic, I have drawn it such that we have information on what happens in each steps, but also what is coming in and what is coming out. And I think we need to be careful when we say efficiency that we mean what the word means. That is, that efficiency does not mean capacity or activity, but it is a ratio of energy that's coming out of the system divided by energy that is going in. So efficiency is a ratio. And we can use this schematic to calculate. We can calculate efficiency of each of these steps by measuring output and input of mitochondria. And we can do this using high resolution respirometry and fluorometry, as well as some other tools that we have and are developing in our lab. And so the thought is that in different metabolic conditions, our mitochondria adapt to change its energy efficiency, and under these conditions, there are secondary consequences of energy inefficient mitochondria that we can quantify. And the hypothesis is that these secondary consequences produce metabolic phenotype that are associated with these metabolic interventions. And today in my talk, I will be discussing 4 examples of the studies that is ongoing in our lab that is looking at how metabolic intervention change mitochondria to make it inefficient. But going in one step deeper is what is the mechanism that alters mitochondrial efficiency during these metabolic intervention? And there are different ideas out there. Other labs have been studying the potential that the post-translational modification of the OXPHOS enzymes may be contributing to efficiency. There are other groups that suggest that the protein import into mitochondria may also be a factor. But our lab is interested in the composition of the inner mitochondrial membrane. So inner mitochondrial membrane consists largely of the proteins as well as these lipids, and they are mostly phospholipids. And like I've said previously, inner mitochondrial membrane sits at the center of the energy exchange processes of oxidative phosphorylation in all 4 steps of the oxidative phosphorylation that I described. And there are 3 reasons why we think that the IMM lipids may be an important factor to regulate respiratory efficiency. First, as shown in this figure, inner mitochondrial membrane lipids can be generated at inner mitochondrial membrane. As many of you may be aware, different organelles from the cell have different lipid composition, and in particular, IMM is highly enriched in 2 types of phospholipid, cardiolipin and PE, phosphatidylethylamine, compared to other organelle. Cardiolipin in particular is a highly mitochondria-specific phospholipid. There are reports that cardiolipin can exist in outside of IMM, particularly OMM, but also in ER. But most of cardiolipin resides in IMM. PE is prevalent in most organelles everywhere in the cell, but it is highly enriched in IMM. And the reason why both of these lipids are highly enriched is because there are these enzymes or series of enzymes. For PE, there's an enzyme called PSD, or phosphatidylserine decarboxylase, that generates PE from PS. And for cardiolipin, there are a series of enzymes such as CLS, cardiolipin synthase, and tafazin, 2 enzymes that I'll be referring to later on. So IMM have mechanism that is resident in its environment to alter its phospholipid composition. So that's number 1. Number 2 is that there are biophysical and structural studies that show phospholipids, and in particular, it's these 2 phospholipids, CL and PE, bound to the enzymes of oxidative phosphorylation. And not only that some of these lipids can alter the activity of these respiratory enzymes. And 3, there are human mutations that are known to cause mitochondrial defects, in particular in these 2 enzymes that I have discussed just now. The PSD and tafazin are their mutations are known to cause a substantial defect in humans. Barr syndrome is a fairly well-known abnormality that occurs as a result of a mutation in tafazin gene. And this is an X-linked condition such that for males, having one mutation in the X gene is sufficient to cause this. So most of these are found in boys. But there is a report that this can also happen in female in a homozygous condition. And there are more recent, and in fact, just last year, two independent studies, one from Canada and another from a group that studied Indian population, that showed that the PISD also is a mitochondrial disease genes. Now, in both of these situations, the reported cases are essentially a functional homozygous null, meaning that these mitochondria do not have an activity to produce cardiolipin or PE. But in our situation, the questions that we are trying to address is the change in the mitochondrial phospholipid that happens in a more subtle way. And so not a homozygous null, but something that is more akin to heterozygous, or something that is more modest. And so, like I mentioned before, today I'll be describing 4 different interventions, 4 different examples, of which that coincide with changes in the mitochondrial membrane phospholipid composition and some gain or loss of function studies that are ongoing associated with these topics. So first, I'd like to talk about the concept of skeletal muscle energy efficiency, and the change that it happens to its efficiency with exercise, but also with disuse or inactivity. So in the field of exercise physiology, the concept of muscle energy efficiency have really existed for a long time. And partly that's because it's been known that the exercise can improve efficiency. And we're not just talking about mitochondrial efficiency here. We're talking about all systems efficiency improve with exercise, such that occur with joint smoothness, with contractile proteins, isoform changes, and so forth. There are changes that are known to happen with exercise in the body that helps improve efficiency. And I think that intuitively makes sense for all of us. When we train or when we do regular exercise, it gets easier for us to exercise. And that is a sign that at multiple levels, our body is becoming efficient. I'd like to highlight the work done by Dr. Carlman Wasserman, who actually passed away a few months ago. And just over 50 years ago, he published a really eloquent and opinion piece coupled by some data that discusses this concept of muscle energy efficiency, both during rest and during contraction. And we all know from our high school physics that delta G does not equals delta H. You have to subtract T delta S. And so every time energy is converted from one form to the other, there's always this portion of energy that is leaked out. And we know in skeletal muscle that for every step of energy conversion that takes place, and in fact, this is for every energy conversion that takes place anywhere in every biological, any system in this universe, there is T delta S that escapes from the system. And so we know that muscle energy efficiency can change. And in fact, here are some data of mitochondrial energy efficiency in skeletal muscle. And on top is the efficiency for electron transfer. So that's the step 2. And C is control and T is trained. And it's shown here that the H2O2 goes down. H2O2 is a metric that we use to quantify electron leak. Electron, when it leaks from mitochondrial oxidative phosphorylation, it reacts with molecular oxygen to produce superoxide. And in our assay, we include the recombinant SOD to push that superoxide production into H2O2, which can be measured with amplex red that we quantify with fluorometry. And so over here, H2O2 going down with training suggests that there is reduced electron leak. And below is mitochondrial P2O. That's what's often referred to as. It's ATP produced per oxygen consumed. And so this is a kind of a combination of step 3, step 4 efficiency, but that also improves. There's more ATP produced per O2 consumed. I like to focus on this section on the improvement of electron transfer with exercise, but actually more importantly, the increase in electron leak that happens with inactivity. And the reason why we're interested with an electron leak increased with inactivity is because this process has been implicated for inactivity-induced insulin resistance. So we're talking about the disuse, not necessarily obesity, but I think the mechanism might be relevant for the same reason I discussed before under the concept of energy mismatch. But regardless, we know that inactivity induces mitochondrial electron leak, and we know from multiple systems and under different circumstances, electron leak leads to oxidative stress that may cause insulin resistance. So we are interested in the possibility that perhaps changes in mitochondrial membrane lipids may induce oxidative stress under inactivity. So for this project that had actually started, and this is one of the only few published data you'll see today, but was led by Tim Heaton, who is a very talented postdoc in my lab who led this study. And just briefly, because these data are published already, what he did was to do a very simple study by exercise training mice for several weeks. And after that, we collected skeletal muscle tissues. And importantly, we isolated mitochondria. So we didn't just do lipidomic on the muscle. We did lipidomics that is specific to mitochondria. And when you do this, on the y-axis, you see UT untrained, T trained. On the x-axis, you see different classes of lipids, and each boxes represent different species of these classes of lipids. He also done this for inactivity, and we used for this study a hind limb unloading model, which is a tail suspension study that make its hind limb inactive. And so you can see the similar sort of layout here, control on the y-axis and HU for hind limb unloading. So that's disuse. And one of the common change that we saw here was a change in PE. So with exercise, the yellow is high and blue is low. So with exercise training, there's an increase in mitochondrial PE, and with inactivity, there's a decrease in mitochondrial PE. So we wanted to know what is the cellular consequence, specifically mitochondrial consequence to mitochondrial energetics when we change PE. And to do this, Tim has targeted an enzyme that produces mitochondrial PE, the PST enzyme that I've described before. And these studies were done in collaboration with Dr. Darren Neufer at East Carolina University, who's an expert in mitochondrial bioenergetic, and really was a mentor to me and to others in my lab when we were located at East Carolina University. And so Tim have generated the muscle-specific PST knockout mice using the tamoxifen-inducible HSA-CRE that was generated by Karen Esser. And as I will show you, he quantified the efficiency for step 2, which can be quantified, again, because we have SOD in this prep. We can quantify electron leak as an H2O2, and because we're talking about efficiency, it's a ratio divided by O2. So the question is, the step 2 of the system, how does its efficiency change when PE is reduced? And again, this is already published, but the H2O2 per O2 is substantially higher in skeletal muscle when mitochondrial PE is lower. And so you can read more about the homozygous knockout data, which has already been published in a Science Advances paper last year. But just briefly, the homozygous knockout mice are lethal, and that's because both the muscle experience quite robust and rapid atrophy, as well as muscle weakness. And so you can see that the mice are kyphosed and the muscles are smaller. And on the bottom, this was the measurement that was taken using the instruments by the Aurora Scientific, but it's a specific force from diaphragm muscle of these mice. So these mice are weak and smaller, and they die. But like I said before, we are also interested in the subtle changes in mitochondrial lipid that happened with metabolic innovation. And so these studies are now led by Pia, a very talented PhD student in our lab, who have been studying the heterozygous knockout mice. And these mice are more prone to insulin resistance. So we are studying whether this mechanism may be important for inactivity-induced insulin resistance. And we're doing that by the loss of function studies as shown here, as well as the gain of function study. We have the muscle-specific tamoxifen-inducible PSD knock-in mice. And so we are using that to study that possibility. But there's actually one technical hurdle to do these experiments. And let me explain why that is before I switch to the next topic. And that is the rodent model for disuse. So, and in particular in mice. So in humans, it's been well documented over many, many studies that inactivity induces skeletal muscle and systemic insulin resistance. This is one example of such study done by my collaborator, Michael Drummond here, and his postdoc, Ziad Mamasani. And I'm not going to go over this, but in this case, it was a bed rest, and the patients become insulin resistant over 5 days bed rest. Now, in mice, there are several models of disuse. High-limb unloading is one of them that I have discussed before. But in high-limb unloading in mice, the insulin resistance is not observed. In fact, in skeletal muscle, there is actually an increase in glucose uptake that occurs both in basal or non-insulin stimulated condition, as well as in insulin stimulated condition. So this is not a good model. The high-limb unloading in mouse is not a good model to study inactivity-induced insulin resistance. There are other methods such as denervation and high-limb immobilization, which also did not really work out for us. And so Pia, as well as Ziad, a postdoc in Micah's lab, have come up with a different method to do this. And this was not necessarily their novel idea, but they came up with a new system that worked with mouse to do this specifically for to induce insulin resistance. And so this is a small cage housing system. The schematic is shown on the left. And what is very nice about this system is that when we put this is a single cage housing, small cage for mouse, but the mice have reduced activity, but they do not gain weight. They do not gain adiposity. They do not have increased fat mass. So going back to the question of what is it about, obesity that is inducing, in this case, insulin resistance. Same thing with inactivity. What is it about inactivity that is inducing insulin resistance? In this case, we can study in the absence of change in adiposity. And the most important point is that with this intervention, the mice do develop insulin resistance, the decrease in delta 2DG uptake. So we are, and specifically really Pia, is utilizing this new model of mouse disuse to study insulin resistance that occur with physical inactivity. Okay, so we're now going to change our topic to another tissue whose mitochondria is highly important. Well, maybe for every tissue, mitochondria is highly important, but for the physiology that we are discussing, thermogenesis in brown adipose tissue, mitochondria is very important. So thermogenesis by adipose tissue happen both in mice and in humans, and the location of the brown adipose tissue, as well as sort of an intermediate inducible thermogenic adipose tissue, which is known as beige adipose tissue. The locations of these adipocytes or adipose pads are shown in this schematic. But like I said, the mitochondria play an important role in brown adipose thermogenesis. The thermogenesis of brown adipose tissue is largely accounted for by a protein known as uncoupling protein 1 or UCP1 that also resides in a mitochondrial membrane, along with these enzymes of oxidative phosphorylation I have discussed. And the UCP1 do so, like I said, by the concept of delta G equals delta H minus T delta S, but really the emphasis here is T delta S, that the proton gradient is used to dissipate heat instead of producing ATP synthesis. Like I said, this occurs across the inner mitochondrial membrane. And so we were interested in the effect that the mitochondrial membrane lipids have on a UCP1 function. And a couple of years ago, there was a really elegant study that came out with my collaborator, Zach Gerhard-Heinz, in the University of Copenhagen. And in that paper, one of the first data that they show is a metabolic profiling of brown adipose tissue across 5 different temperatures. And in this paper, they have found cardiolipin to be one of the metabolites that is most highly affected by cold as well as thermoneutral condition. And so they generated a loss of function mouse. They also studied gain of function in vitro to show that if mice do not have cardiolipin in white or brown adipose tissues, then these mice are not capable of producing as much heat. So these are cold intolerant mice. And they did so again by targeting the enzyme cardiolipin synthase, which is an upstream enzyme, the first committed step that makes the first cardiolipin molecule. So one of the things that we learned when Tim was doing the study for muscle mitochondrial lipids is that we need to isolate the mitochondria to do this, or to get the whole picture at least. And that's because many of these interventions that induce changes in energetic demand also change the abundance of mitochondria. And so if you don't normalize the mitochondria, you may be seeing increase or decrease in lipid content simply because there are more mitochondria or less mitochondria. So Jordan, a PhD student in my lab, who's actually defending his dissertation in a couple of weeks, he has done this by isolating brown adipose tissue from mice and isolating mitochondria from these brown adipose tissue. And similar to Zach's study, we have done this in multiple conditions, and this one is in the cold. So as you can see, CL is a third one that's shown on this, and these heat map are shown not are not showing all the species, but all the species per class of lipids are combined, and CL is significantly higher. And again, this is normalized to mitochondria, so it's different, but it supports the notion published by Zach's group that the cardiolipin is important for brown adipose thermogenesis. And Jordan also did this in the mice that were housed in thermal neutral condition, and this was a little bit surprising. And so if you see here, cardiolipin is actually not different if you normalize to mitochondria between the mice that are housed in room temperature. But PE was actually lower. So when mice were put in a thermoneutral condition or a condition to brown adipose mitochondria where there is decreased energy utilization, then the PE goes down. And in cold, both PE and cardiolipin goes up. So Jordan did studied another condition. So similar to thermoneutral, we use the UCP1 whole body knockout mice. So these mice are similar to thermoneutral mice in that they do not have the mitochondrial energetic burden. And in fact, the mitochondrial lipid portfolio in the UCP1 knockout mice is very similar to thermoneutral mice and thermoneutral wild type mice. And as you can see here, PE is down in UCP1 knockout mice, just like in the thermoneutral mice, and no change in cardiolipin. If anything, it's going up, which is the opposite direction what you expect. So we have collaborated with Zach's lab to study the role of mitochondrial PE in brown adipose tissue thermogenesis and in mitochondrial energetics. And we have done so by using the flux mice that we discussed before, now crossed with the UCP1 ERT2 or tamoxifen-induced UCP1 specific Cree mice that we got from Christian Wolfram, who was the 2 speakers ago from this obesity webinar. So we got these mice from Christian and crossed them to the flux mice. And as you can see on the right side, these mice are highly cold intolerant. And Jordan's been doing the mitochondrial phenotyping of these mice, and we are showing some data here. And on the left, what you can see is that the UCP1 dependent oxygen consumption is lower, but the ATP synthesis, the rate of ATP synthesis is the same. So I forgot to say that the VO2 is lower in this mice and JO2 in mitochondria is lower, and that's explained by lower UCP1 respiration and not by lower ATP synthetic rate. And another thing that we notice in this brown adipose tissue, consistent with the reduced energy consumption, is that they are wider, and these are explained by larger lipid droplets shown in histology. So going through more quickly, I'll present 2 more different interventions that we have been studying. The first of these is in the liver, and it's specifically in the context of non-alcoholic fatty liver disease, or NAFLD. NAFLD is a progressive disease strongly associated with obesity that is triggered by initially a lipid accumulation in hepatocytes that produces what's shown as a second step of this, steatosis or steatotic liver. And steatotic liver is simply hepatocytes with more lipid content that is not necessarily pathologic yet, but they can produce, rather progress to subsequent steps undergoing fibrosis, necrosis, and some of these patients can experience hepatocellular carcinoma and even death. And so there are interests to suppress the progression of NAFLD earlier on to prevent the progression of the steatotic liver into further steps that are pathologic. And so one of the things that is known about the progression of NAFLD is in the mitochondrial oxidative stress. And so we wanted to know what happens to hepatic mitochondrial lipid composition with NAFLD. And so this project is led by Marissa Lang, who's a PhD student in my lab, and we're collaborating with Scott Summers here at Utah, and specifically with his student, Annalise Poss, who have been doing these studies in the liver. And so just like Jordan and Tim studying in brown adipose and in muscle, we studied several models of NAFLD and steatohepatitis in wild type mice. And without going into too much detail, what we found is one factor that is common for all these 4 interventions is a reduction in mitochondrial cardiolipin content. And so we collaborated with Zach because he generated a cardiolipin synthase flux mouse, and now crossed this with albumin Cree to study the effect of low cardiolipin in hepatocytes. Now, earlier this year, a group in China have published a mouse that we are also studying. They've generated their own flux mouse, but they have found that the cardiolipin synthase that is specifically knocked out in liver, these mice have fatty liver and they develop fibrosis. Now, they attribute the changes in these phenotype to a transcriptionally mediated mechanism. Zach also have data about that in brown adipose tissue, but specifically, Marissa is interested in the effect that the loss of mitochondria have on mitochondrial bioenergetics. I will just briefly say that even in the absence of high-fat diet, so these are in standard chow-fed young mice, these cardiolipin synthase liver-specific knockout mice have fibrosis, as shown here by trichrome blue, as well as an EM image. And she's been doing the oxphos phenotyping on this, and this is a step 2, similar to the muscle story that we've described, and there's an increase in the electron leak. So a decrease in efficiency of step 2 as a result of reduced cardiolipin in hepatocytes. So these studies are currently ongoing, and I think it's so far very promising. So in the last few minutes, I'll talk about the last of the projects, which I am very excited about. And this is on the concept of weight loss and how it might change energy efficiency of the body. And so a group led by Rudy Leibow has been saying this for some time, and that when a person undergoes weight loss, that their energy expenditure, systemic energy expenditure goes down, or their energy efficiency goes up. And this may be one of the many reasons when a person loses weight, or a mouse for that factor, when they lose weight, they may be more prone to gaining their weight back. And in their studies, as you can show here, their primary data shown here, one of the data on the right shows a condition with a 10% weight loss, and on the left is the same individual before the weight loss. And you can see that the 24-hour energy expenditure is lower, resting and non-resting energy expenditure is lower. And I think it's particularly interesting that the non-resting energy expenditure, or exercise energy expenditure, is low, because that suggests that the skeletal muscle, as many tissues increase in its energy expenditure during exercise, but a lot of that comes from skeletal muscle. And so we were interested in the possibility that the weight loss is increasing skeletal muscle energy expenditure, or rather skeletal muscle decreasing skeletal muscle energy efficiency. So a postdoc now, Patrick, who have just recently graduated with his PhD in my lab, now doing a brief postdoc in Micah's lab during COVID. Together with Marissa, have done a very simple intervention in wild-type mice, where we have placed them all on the high-fat diet feeding, and then half of that was then separated and removed, put back on standard chow. And when we did this, an interesting thing happened, and that is the weight loss shown by WL here lost body weight for the first couple weeks, and then for the next 3 to 4 weeks, their weight stabilized. So I think this is a nice observation, but it's an important point to keep in mind. And so these measurements were done not when they were actively losing weight. And I think that's, again, an important thing to keep in mind when we study obesity. What is it about obesity? What is it about weight gain, weight loss? And in this particular point, the mice were no longer changing weight. And we measured the whole body VO2, and consistent with Rudy's data, the energy expenditure normalized to lean mass was lower in the weight loss group. And so we studied skeletal muscle energy expenditure in these mice. And briefly, now we're talking about the efficiency for ATP synthesis. So this is a combination of step 3 and step 4, quantified by P2O, or the ratio of the JATP to JO2. And that goes substantially higher in mitochondria from skeletal muscle in the weight loss group, about 60% increase. And I think this is quite substantial, quite meaningful. Again, I want to remind you, this is in the wild type mice, not genetically modified mice. There is a substantial increase in skeletal muscle mitochondrial P2O that coincides with weight loss. And so, of course, we have done the mitochondrial lipidome in these, and we are focusing on this one specific lipids, TLCL or tetralinoleoyl-cardiolipin. And I will tell you why this rather than just pan-cardiolipin that we're studying in liver. Now, cardiolipin, just briefly, has 4 fatty acids of 4 acyl chains, and tetralinoleoyl-cardiolipin is an 18-2 for all 4 of the acyl chain of the cardiolipin. And this is increased by 2-fold in a weight loss group. And why we're interested in tetralinoleoyl-cardiolipin or TLCL is for these data. Now, TLCL is generated by an enzyme tafazin, which I've described before, in an enzyme that is defective in patients who have Barr syndrome. Step 4 over here is the cardiolipin synthase. Cardiolipin synthase produces a 4-acyl chain cardiolipin, but for a not very clear reason, we and others think that this cardiolipin is not functional. And we don't really know why that is, again, very well anyway. And we call these premature cardiolipin, and it goes through transacylation process to sort of knead out the cardiolipin legs into 4-18-2, which we call mature cardiolipin, which is TLCL. And so you can see that this enzyme is terminal. If you don't have it, you will still have the premature cardiolipin, yet the patients with Barr syndrome have severe defect. And so there is something to making the cardiolipin all 18-2. Anyway, we have studied these mice before, and one of the things we've seen, and this was not the point of this paper that was published by Jordan, who's the PhD student I described before in the brown adipose project. These mice are less prone to developing obesity, and they have increased energy expenditure. And so we are interested in the possibility that the TLCL is the important lipid that regulate mitochondrial P2O. There are some other data, not by other groups, that suggest that the cardiolipin may be a regulatory factor for P2O. And I'm happy to discuss that later if any of you have questions. And so I am wrapping my talk now. In summary, our lab, as you saw in the 4 examples that I presented today, we're interested in mitochondrial lipids as a fundamental mechanism, a common mechanism that regulate efficiency across multiple cell types / tissues, and that the mitochondrial lipids change with metabolic interventions, and these changes may induce secondary consequences to mitochondria that induce pathology across multiple tissues. So I'd like to thank the people who are involved, those in my lab as well as our collaborators. Those shown in red played a major role for the projects that I presented. I'd like to specifically thank my collaborators, Daryl Neufer at East Carolina University. Our lab was in ECU for several years before we relocated to Utah, and Daryl was a huge part in our success. He really changed how I think about mitochondrial bioenergetics. Really, I didn't think much of mitochondria rather than a place that produced ATP, but he really was my mentor at my time at ECU. So thank you to him. Also, Zach, he has been a great fun to interact with. We actually have, over the pandemic, started a monthly Zoom lab meetings where we present data, and it's just been really a fantastic way to interact. And also Dennis and Jean Vance, who have been my mentors since I have become an independent PI now 7 years ago. And they are phospholipid experts, and they have really helped me find my identity in this path of scientific endeavor. So, and thank you all of you for listening, and I'm happy to take questions that you might have.
And so let's jump right in. First question today. So Katsu, you talked about how mitochondrial inefficiency might have a detrimental effect to health, but it seems like in the weight loss portion of your talk, the increased inefficiency would increase energy expenditure to prevent obesity. Could you just clarify your thoughts on that?
Yeah, sure. So I didn't touch on this very much, but so the concept of whether increased efficiency or decreased efficiency is, beneficial, quote unquote, is highly tissue dependent and highly step dependent. And I think the highly step dependent is quite intuitive because the signaling cascade that is induced by such inefficiency will be different. Now, tissue dependent. So in the context of what you just asked about weight loss, and so weight loss increases energy expenditure. Sorry, weight loss increases energy efficiency, decreases energy expenditure. So from the context of adiposity, weight gain, the fact that if skeletal muscle has an ability to use more energy to do the same work, that is decreased energy efficiency, not with weight loss, then that will be predicted to be beneficial from the context of obesity, but not necessarily if you need as much energy to produce force in the case of skeletal muscle. In the case of cardiac, though, for example, if there is a reduced efficiency, that may be potentially highly detrimental, both from the context of contractility as well as malignant hyperthermia. So that is a situation where decreased P2O will be bad. Another tissue where this could be bad that we're actively studying is in a beta cell, where an ability to sense the ratio between ATP and ADP is the intermediate signal to release insulin stimulated by glucose. And so if mitochondria is inefficient in beta cell at this step, then it will also be predicted to be ineffective insulin producer. So that's also not good. So again, it's highly tissue dependent and highly step dependent.
Excellent. Great answer. And now another question here. In the last portion of your talk, you discussed how weight loss might increase the efficiency of for ATP synthesis. How does exercise alter efficiency, and how does that relate to exercise and its relationship to gaining weight?
Right. Okay. So that's a really good question. Now, so it's not our data, but I showed it earlier that exercise actually increases the efficiency of ATP synthesis also. So that's the step 3 to step 4. P2O is increased in P2O. And so that would be expected to reduce the energy expenditure with exercise, right? And I think in the context of weight loss and exercise, there are two things going on. So if energy efficiency is higher, then energy expenditure per ATP produced is lower. But if a person is exercising, then the total ATP consumption is higher. So maybe the muscle is potentially more efficient and less effective user of energy, but it may the muscle is utilizing a lot of energy to begin with. And the reverse is also true. When a person is inactive, then the efficiency may be lower, meaning energy expenditure per ATP consumed might be higher, but that person is not exercising as much. So the non-resting energy expenditure is lower to begin with, and therefore the low efficiency may not be beneficial from the context of adiposity.
Excellent. Here's another question here. What is known about IMM cardiolipin and PE levels in obese humans and mice?
So the question is mitochondrial cardiolipin and PE content with obesity, right, in mice. So again, it's
Yes.
tissue dependent. We presented the liver data, right? The obesity increases, obesity decreases cardiolipin. I'm trying to remember. PE went down in some intervention and PE went up in some intervention. And that's why we're not particularly focusing on PE at the moment in the liver with obesity. We also looked at muscle, and so there are a few things that are a caveat to this. So when you induce intervention, metabolic intervention, I mentioned that one of the things that you need to be careful is that the mitochondrial content changes. Now, one of the other things that also changes is actually protein to lipid ratio of mitochondria change. So you need to be kind of very careful how you think about this. You want to usually when you do lipidome, you normalize it to the milligram of protein of mitochondria. And if you only look at the data that way, then you miss the picture because the protein to lipid ratio is also different. And this protein to lipid ratio we find is actually kind of highly plastic, and it's really highly time dependent. So if you look at it over different course of time, this concentration is different. And so it's quite tricky, and it's not simply sort of one goes up and down. You have to, the question is, what are you comparing to? That is always important. And I think in my mind, what's important is compared to the rest of the IMM environment. Yes, I think protein to lipid ratio is also important, but you want to think about the lipids that's present. And if cardiolipin is not there, then you have PE or PC or something else that's there. And so it's all about sort of the ratio. And I don't have a great answer to this without showing you sort of all the data here, and we have it for muscle. We also are doing this for white adipose tissue. That's another tissue where obesity obviously directly is important. So we have to troubleshoot to make sure we can get enough mitochondria from a different white adipose tissue to do the lipidome, and we got that now. So I know I didn't really answer your question, but in the tissues that we measure, they do change, but the change is not sort of simple.
Excellent. Perfect. Well, yeah, that was a great answer. And I think in the interest of time, we'll just try to squeeze one more question in here. The question being, is the muscle atrophy with PSD knockout just a loss of ATP bioenergetics, or does it cause mitochondria signaling for apoptosis or autophagy or mitophagy?
Yeah, great question. And so one of the things that, and we discussed this a little bit in Tim's paper too, there is a mitochondrial defect. JO2 is lower, JATP is lower. But the magnitude, I think it's kind of interesting. The magnitude, it is not the extent that you might actually expect what we saw, which is death. So the magnitude of loss in JATP was probably like 40%, and that is capacity, right? And so when you have a 40% loss in capacity for ATP synthesis, do you expect atrophy? Well, I'm not sure. Maybe atrophy, yes, or at least partly explain it. But we can live with a 40% reduction of mitochondrial mass and muscle for most of the function, because at basal state are about 30% of the mitochondria is only active in muscle. So you would expect that with the 40% reduction, so you still have 60% reserve, that would not necessarily be sufficient to cause the phenotype we saw. So the bottom line is we really don't know why the PSD knockout mice and muscle knockout in mice were that severe. We have some lead, but I don't think it's JATP.
Excellent. Well, Katsu, thanks so much for all your fantastic insights today, both in your presentation as well as the Q&A session.
Yeah, thank you, Liam, and thanks for the opportunities that you guys provided, as well as also for all the audience that have listened in, and hope you guys stay safe.
And in closing, thank you again for taking part in this Inside Scientific webinar produced in partnership with the American Physiological Society and we look forward to having you with us again soon.